Hello, Newcomer biology students. Welcome to the Osmosis virtual lab. I'm Mr. Fonseca, you know me, and below me you can see that that's Miss Galusha. She is the science coach for your teachers in the science department. Both of us are here today to help you understand how to do this virtual lab for osmosis. So there's a few parts. Follow along with us. The first thing you'll do is you're going to click on that link, and that link is going to take you to this website. This website is where you're going to be doing your osmosis lab. The thing you're going to do on that website is you're going to take a look at the objectives. The objectives for the lab are right here on the left-hand side. You're going to have to scroll down to be able to see the objectives. All right. Once you see the objectives in this document here that you're working on, you're going to be filling in the blanks for the objectives. So the first one, describe the process of, let's look on the site. Objectives describe the process of osmosis. So osmosis is what you'll be filling into that blank. Okay, go ahead, read the objectives, and fill out these blanks. Pause the video now so that you can complete this part. All right, great. So now you've paused the video, you've completed part 3, and you're now going to watch as we do the procedures together, and then you're going to try this on your own. So part 4, procedures. The first thing we're going to do, as I'm reading the directions, is select the red blood cell and drag it into one of the 3 beakers. So here's my red blood cell. I'm going to click on it and drag it and put it into the hypotonic beaker. I saw something change when I put it in there. What's step 2? It says observe the process of osmosis. Determine if water moves into, stays at equilibrium, that means the same or balanced, or moves out of the cell. Observe what happens to the shape and size of the cell. So water, I can say water moved into that cell because when the cell was outside, it was flat like this, and then when I moved it into the hypotonic solution, it got big, full of water like that, so water moved into it. Okay? And it's asking me about what happens to the shape and size of the cell. So before I put it in the solution, it was flat like this. After I moved it into the solution, it got bigger and round. So the shape changed to become round instead of flat. And it got filled with water. It's bigger than it was before. Right? So I would record, so step 3, record your observations in the table below under data. So our table, if we scroll down, is right here. Let's see, red blood cell, hypotonic. It says in a hypotonic solution, water moves blank of the red blood cell. So in a hypotonic solution, water moved inside of that red blood cell. So here, I'm going to put inside of the red blood cell. And then also for hypotonic, it's asking me, in a hypotonic solution, the cell looks, how does the cell look? Well, like we talked, the cell looks round and bigger than it was before. So I'm going to say the cell looks round and bigger than it was before. So I've talked about how water goes into the cell, and I've made an observation of what the cell looks like. And I did that for the hypotonic solution. Now it's your turn. You're going to do the same thing. You've already copied the notes down for the red blood cell and hypotonic. You're now going to take that red blood cell and try putting it in an isotonic solution and make observations of what happens and record those on your data sheet. And then in a hypertonic solution, make observations, what happens, put them in your data sheet. And then you'll do the same thing with the Elodea cell, observing it in the hypotonic solution, the isotonic solution, and the hypertonic solution, and again writing your observations of what happens with the water going in or out of the cell and how the size and shape change. And last, you'll do that with the paramecium. in the hypotonic, isotonic, and hypertonic solutions and in your data table, making observations about what happens to the cell. Does water go inside it or outside of it? And how does it look after you put it in each of those solutions? After you finish with that, the last thing you'll need to do for this lab is answer this conclusion question.